Hello there, and welcome back to Hydroponics Daily. I mentioned in a previous post about chimeras and how they can be one of the ways variegated plants can arise. However, I said there are lots more interesting examples of chimeras in horticulture, and I wanted to do an episode all about them. And I thought this would be a good time to do it because I like to do a interesting episode about cultivation of plants and then do one about problems or challenges, things like that, or techniques, just to split it up. Because if it was just like pests, diseases, problems, physiological disorders, or equipment, it would get quite dull. So let's mix it up and have an interesting one about how these obscure plants arise and some of the examples of them. I've got 3 really good examples. So he said that a chimera is named after the Greek mythological monster of the same name that possessed the head of a lion and the body of a goat and the tail of a dragon.
I can use ChatGPT to answer that. You can enable ChatGPT on your iPhone.
No, you're okay. I don't want ChatGPT. My phone has just decided to have a life of its own.
No problem. I won't.
Good. I don't know why that came on, but yeah. Welcome to my world where ChatGPT is trying to take over. You can tell from that this isn't a fake AI voice that's just reading a script from a AI-generated prompt, because I can keep in nonsense like that. So I said chimeras, they are plants that are actually separate genetic materials that have been mixed together, and they keep their genetic integrity in the individual cells, but different cell lines create them really strange looks. So he said the chimera, a head of a lion, a body of a goat, a tail of a dragon, and a hypothetical chimera plant would have the flowers of a tomato, the roots of a potato, and the stems of deadly nightshade. There's an example, keeping the genetics the same, but different cell lines, different cell structures. So they are very, very rare, and they occur on fortuitous occasions in plants. They are not something you can pre-program. They're not something you can genetically engineer. If you could come up with a way of doing this, you'd be a rock star in horticulture and plant science in general, and you would earn a shed load of money because you'd be able to produce so many wild and wacky different ornamental and edible plants. They normally form when a graft union, so when you try to graft a plant, put a scion on top of a rootstock, that somehow the cells get mixed up in the graft union, and then a stem appears that has got a mix of both the rootstock and the scion cell lines. So it might be the xylem and the phloem are from the rootstock, and the bark is from some of the top bit. So, and then if you can propagate them from cuttings, obviously you can't propagate them from seeds because they just go back to one single cell line. And the mixed plant altogether is called a chimera. They can occur in animals, and you can get little rats and mice that have got black fur and white fur mixed together, and that's because they are like twins that have been mixed together, one black head, one white head. And it can even happen in humans, and you can have problems where one of the sort of like the twin organ has issues. So they're normally deleterious in animals and humans, but in plants they can be wacky, just like all mutants can. Like, mutants in horticulture can be prized as unusual. This is not a mutation, but it is a developmental disorder, should we say. They're not happening as it should be, so you can call it a disorder. The first report of a graft chimera came in 1674, and that's crazy because that predates any knowledge of genetics and plant breeding. 1674, so that's way before Mendeleev, the Polish monk who discovered, the way genes segregated and things like that. So Pietro Natti, he was an Italian scientist and the director of Pisa Botanical Gardens. Pisa Botanical Gardens is the oldest botanical gardens in the world, I believe. I'm not reading that from a script, and from memory, so it might be wrong. But I'd love to go to Pisa Botanical Gardens. Most people, when they go to Pisa, they want to go and see the Leaning Tower. I'd rather go to the Botanical Gardens and see where Pietro Natti first discovered a remarkable chimera, which was not cross. You don't say cross because it's not a cross. It's a chimera. It's not hybridization. This chimera had sour orange cells and citron cells. So a cross between two different citrus species. And that would be, quite difficult to note because they're both citrus. But the most famous example of a chimera that resulted from a graft union is called Adam's Laburnum. So if you know laburnum, you know they have bright yellow flowers. This plant is a combination of laburnum, anogyroides, and a broom. And brooms they're both in the bean family, or the bean family, the Fabaceae. But these are completely different species, but also in completely different genera. So laburnum, anogyroides, and Chymocytus purpureus. So that's purple. One's got purple flowers, one's got yellow flowers. And because it's still essentially two species joined together, they cannot have conventionally scientific species names. Instead, Adam's Laburnum, it has a little plus before the genus name. So it's plus Laburnocytisus adamii, and the plus indicates its unusual origin. And the fact that the Graft Chimera are still two distinct species is also the reason scientists refrain from using other gardening terms for them, like mosaic hybrids. Mosaic hybrid sounds like it's sort of like got a virus. There's a number of different ways of writing the species as well. So the majority of the reason why Adam's Laburnum is so interesting, because for the majority of the branches, the laburnum is the dominant influence. But the epidermis, the skin, is composed of cells of the broom. So it's easy to spot when it's in flower because the flowers of both species form on the plant. And so you can have yellow and purple flowers on the one plant. And laburnums have this mass of yellow flowers normally. So you get a mass of yellow and purple flowers. So that's really interesting. Another chimera that results from graft union is the Dallimore's chestnut. And that's called Aesculus plus domestica. And if you see these written down, these scientific names, you'll notice that the plus sign indicating that it's a chimera is in the middle between the genus and the species, whereas in Adam's Laburnum, it's before the genus name. So you know that is an intergeneric chimera, whereas this one, Dallimore's chestnut, is a chimera between two chestnut species. So it's a less obvious graft chimera, as the species providing the tissues in both are chestnuts. What can you say about this? So the chimera is a fusion of cells from the yellow buckeye, Aesculus flavor, and horse chestnut. I think Aesculus buckeyes are there from like Ohio and places like that. I'm pretty certain isn't one of their universities named the Buckeyes. And then horse chestnut, I think that is from the Balkans. So completely different geographically. It was discovered in Kent in the UK in 1955 by William Dallimore, a retired director of Kew Gardens, the largest botanical gardens in the world. And the tree is characterized by having misshapen, crinkly leaves and flowers that appear later than the common horse chestnut. There are 3 different types of Dallimore's chestnut. Despite the unusual ornamental features, they are very rare in cultivation. So if you can find a Dallimore's chestnut, then you're doing really well. Take a cutting, if you can, and try and propagate it, because it's a very unusual plant. And then a third graft chimera that can be grown in gardens is a fusion of cells from a hawthorn. So if you know what hawthorn is, I'm not going to describe it. I can't describe a hawthorn. Flowers, it goes into leaf really early. It's got thorns on it. It's also called mayflower. And where I live, they've got purple flowers, but most places they've got white flowers. Because I live in the Midlands of the UK, and the Midlands hawthorn has got pink flowers. And then this graft chimera is a cross between hawthorn and medlar. And medlars are popular down in Kent, near Dover, on the way to France. Medlars are unusual because it's the only fruit that you have to rot before you eat it. So you allow them to rot before you eat them. And the only place you used to be able to buy them outside of Kent was Harrods in London, because they were so difficult to get and so expensive to get in this rotted state. So the name for this graft chimera is Plus Cratago Mecephalus, which means it's an intergeneric chimera, so very unusual. So although these are 2 separate genera, they do appear closely related, with them also forming a hybrid. So you can get, as well as the chimera, you can get a hybrid. So This one, the hybrid, would have an X on the scientific name, if I haven't lost you all way before this. So that would be X Cratago mesipilis. So you can get a hybrid of medlar and hawthorn, and you can get a chimera. Unfortunately, both the chimera and the hybrid are rarely cultivated, and possibly because the fruit needs bletting, rotting. Bletting is a term for the controlled rotting of fruit, and that removes the sort of astringency of the fruit, makes it much more sweeter. And the idea of eating partially rotted fruits is not a feature that appeals to many people. I've got a medlar in my garden, and I have never eaten it because it's either rock solid or they've rotted to the point where they just look disgusting. So I just grow it for its nice flowers, and it's a really good pollinator. So that's graft chimeras. They are very rare. They would be exceedingly difficult to attempt to purposely grow, but you can actually create them. So one simple graft chimera that can be achieved is by taking the eye out of a red potato tuber and placing it in the hole made by a white potato tuber that has had the eye removed. You then need to remove the other white eyes from the potato and plant as you would any other seed potato. The resulting plant will produce red, white, and blotched and pink potatoes. So that is one graft chimera you could do in your hydroponic grow today. Take a white potato variety, get an eye off a red potato variety, and insert it into where there was an eye of a white potato on a white potato. Take all the other eyes off that potato, that seed potato, and stick it in the ground and grow it, and you'll get blotched and pink and mixed and swirled potatoes coloured. That'd be strange, wouldn't it? So you could achieve that. If you can think of any other chimeras that you would like in an ideal world, or other examples of chimeras, or ways you think you might be able to create them artificially, then please get in touch. I'm more than happy to do a follow-up episode. A number of chimeras result from mutations in cells in the apical buds of plants, and there you can get like a mutant and the wild type growing together. So they're not mixes of two different genetic materials. Well, they are, but it's just a mutant and a non-mutant. So this is where the one cell gets mutated and then gets propagated vegetatively. So two good examples of that are the fernleaf beech and the thornless blackberry. The fernleaf beech produce crinkled leaves due to genetic differences in the epidermal cells. If the bark is damaged, stems without the epidermal cells will grow out, and thus the new stems bear normal classical beech leaves. These normal stems then need to be pruned off to maintain the tree in its chimeric and ornamental state. And the thornless blackberry, or Corrie thornless, is also a result of mutations in the epidermal cells, and this mutation prevents thorns developing. So that would be quite nice, a thornless blackberry, much easier to harvest. You grow a crop that's got thorns on it, like raspberries, rose hips, what else? I'm trying to think of thorned crops. There are quite a few. You could remove them using this. But like the fernleaf beech, the mutation is fairly unstable and needs to be enforced regularly by pruning the thorn stems that arise. So they're the most common chimeras, graft chimeras and bud mutation chimeras. As I mentioned a couple of days ago, when it comes to mutations in apical buds leading to chimeras, it's normally a variegated plant that's the most common type of variegation that is produced and propagated for ornamental purposes. So hollies are a great example. Lots of the variegated hollies are chimeras and can't be propagated through the seeds of the hollies. And weigela and cornus dogwood, they're prone to reversion as well. So hollies are stable, whereas weigela and cornus, they're prone to reversion. I'll give you my two of my best horticultural jokes now. How do you identify a dogwood? By its bark. What's special about its bark? It's rough. Yeah, cornus, the dogwood. Two of my worst jokes, or my best jokes, depending on what you think. And when we say reversion, it's the loss of the mutated cells and the reappearance of the wild types. They just overtake them because they're growing faster, they're photosynthesizing. So reversion is very common in lots of chimeras and mutants and variegated plants. So you need to remove it to keep the ornamental features of your chimeras. Normally, when it's food crops, we think of slower growth of variegated or chimera plants as being a disadvantage. But if you've got ornamental plants like buddleia or rhododendron, you might actually want them to be slower growing. So you can get variegated forms of buddleia that will grow far, far slower. You can even get variegated Japanese knotweed, which is available, but massively invasive and problematic. Even variegated Japanese knotweed would be a massive issue. I don't know about in America, but in the UK, it's a highly invasive weed. You can't get a loan from the bank, a mortgage, if a house has got Japanese knotweed. And it's even a massive weed in Japan. People say, oh, it's not a weed in Japan. We must go and study why it's not a weed in Japan. I've been to Japan, and I know that Japanese knotweed is a massive problem over there. So don't listen to people who say that. So yeah, and you can get reversion in slow-growing plants, slow-growing conifers and things like that. So what else can we say? While graft chimeras are plants containing a mixture of cells from different species, it was found in 2014 that the cells of the graft union of some plants can swap genetic code and even hybridize. So that's classical science says that can't happen. But in 2014, it was found that they can actually swap genes and sort of do full hybridization or certainly partial hybridization. Now, these hybrid cells would normally remain in the graft union and do not form the new plant because they just stay there. They're not being propagated. They're not in the apical meristems that produces the bigger stem and more leaves and things like that. But scientists from the Max Planck Institute in Germany found that they can extract the hybrid cells and grow them using micropropagation techniques to produce new hybrids that before would not have been possible by conventional interbreeding using flowering. So if you've ever thought of a hybrid and you've read that these plants can't be hybridized, even though they are closely related, then maybe you could try and graft them and then extract the cells from the graft union, micropropagate them, and you've got a hybrid plant. Wow. If you're a top-level micropropagator, if you're a keen breeder, then I've saved some really juicy, good stuff to the end of this episode. If you found it useful, if you thought that was interesting, if you thought my jokes were hilarious, either give me a review or share this podcast with somebody else. And if somebody has shared this podcast with you, you owe them a very large drink down the pub. So go off and buy that person a drink. Thank you very much for listening. Until tomorrow, where I'll talk about probably some sort of pest or disease, probably something like downy mildew, because we've not talked about that yet. And that's a really bad disease. So we'll talk about that tomorrow. Thank you very much for listening. Goodbye.